ID 2503108. Our other group member ID is 2503105, 2503106, and 2503107. Our presentation topic is heat shock genes in fish. Introduction to heat shock in fish. HSGS encode stress inducible proteins called heat shock
স্লাইড
proteins.
মুটেও যাও না পরের স্লাইডেও যাও না, হয় স্লাইড মুটে যাও নাহলে পরের স্লাইড দেখাও, নয়তো ঠিক আছে 2 নাম্বারটা দেখাও। ঠিক আছে।
Introduction to heat shock genes in fish. HSGS encode stress-inducible proteins called heat shock proteins, found in nearly all organisms, including fish. They act as an internal defense system that switches on during temperature extremes or environmental stress. Fish are ectothermic, so body temperature follows water temperature, making HSG expression vital for survival. Studying HSGS helps predict how fish population respond to a changing aquatic environment. Next topic is environmental and aquaculture stress factors. Temperature fluctuation disrupts protein stability, quickly triggers HSG activation. Water pollution, heavy metals and pesticides chemically stress proteins. Hypoxia, low oxygen forces anaerobic metabolism and oxidative stress. Salinity change, osmotic shock during freshwater, saltwater migration, overcrowding and handling. Major stressors in farm conditions. Our next topic is classification of major heat shock protein families. Small HSPs 15 to 30 kDa prevent protein aggregation early in stress. HSP 60. Mitochondrial chaperone folds imported proteins. HSP 70. Most studied family in fish, primarily stress biomarker. HSP 90. Stabilize signaling proteins and hormone receptors. HSP 110. Large chaperone that works with HSP 70. Structure and activation of heat shock transcription factors. HSFS are the master switches of the heat shock response. HSF1 is the key stress responsive regulator in fish. At rest, HSF1 stays inactive as a monomer bound to chaperones such as HSP90. Heat stress causes HSP90 to release HSF1, freeing it to respond. Freed HSF1 monomers assemble into an active trimer and enter the nucleus. The trimer binds heat shock elements on DNA, switching on HSG transcription. Biochemical mechanism of action in fish cells. As an ectotherm, a fish's body temperature rises with the water, quickly causing cell proteins to unfold. This unfolding releases and activates HSF1, switching on HSP genes. The resulting HSPs refold or clear damaged proteins, restoring the cell within hours. The next topic is ATP-dependent chaperone cycle of HSPs. HSP70 binds a misfolded protein while carrying ATP. Co-chaperone HSP40 stimulates ATP hydrolysis to ADP. This closes the chamber lid, letting the protein fold safely inside. A nucleotide exchange factor triggers ADP release and fresh ATP binding. The lid reopens, releasing the folded protein, ready for another cycle. Significance of HSGS in fish adaptation and survival. HSPs protect essential proteins from irreversible thermal damage. Fish with strong HSP induction survive extreme temperatures better. Mild pre-exposure builds cross-tolerance to multiple stressors. HSG expression allows fish to acclimate to new or changing habitats. Applications of HSG as bioindicators in aquaculture. HSP expression lets farmers detect stress before visible symptoms appear. HSP levels indicate water quality and pond conditions quickly. Breeding programs use HSP profiles to select thermally resilient stock. Combined with other markers, HSGS help screen for disease susceptibility. Tissue-specific expression of HSGS in fish. Liver and gill show the highest HSP70 induction under stress. Kidney shows moderate expression linked to osmoregulation. Muscle and brain show lower induction. Brain HSPS aid neuroprotection. Gonad tissue shows the weakest response, affecting reproduction. Impact of climate change and global warming on fish response. Ocean and freshwater temperatures have risen steadily over recent decades. Chronic HSG upregulation from constant heat may exhaust the stress response. Altered HSG expression affects metabolism, growth, and reproduction. Stenothermal species face the greatest risk of decline or range shift. Nutritional modulation and immunological roles of HSPS. Vitamins C and E, selenium, and omega-3s support HSP synthesis. Poorly fed fish show a weaker, slower heat shock response. HSPs also act as danger signals that alert the immune system. They help activate macrophages and support pathogen defense. HSP boosting feed additives are now used to improve stress and disease resistance. Conclusion and future research scope. HSGS are indispensable safeguards that let fish survive extreme environments. Future work should expand genomic and transcriptomic profiling across species. Selective breeding and HSP-based biomarker kits aid sustainable aquaculture. This knowledge will be key to climate-resilient aquaculture and food security. Thank you, everyone, for hearing our presentation. Thank you, everyone.